Hello, good morning and good afternoon to everyone. We've got a few people, coming up, so we are about to start within a couple of seconds just to see if everybody is getting on board on time. Thank you. It's 5:01, so we've got more people coming up, but we will start right now. So, as I said before, good morning and good afternoon to everyone. Welcome to our webinar about cancer immunotherapy and tailor-made humanized mouse model solutions. The webinar will be run by Caroline Mignard. So a few information before getting started. So Caroline will run the webinar and we will collect all the questions throughout the webinar and answer them at the end. So don't hesitate to take some notes and ask your question as soon as you want. If you are also facing any difficulty, don't hesitate to ask me on the chat bot on the right-hand side of the window. So, for today, I'm Florence von Bertas. I'm the head of marketing and communication at Oncodesign, and I will be the moderator of the session. So before stepping to the core topic of the discussion, let me introduce you our speaker. So Caroline Mignard has obtained a PhD in biology from the University of Strasbourg in France, and has more than 15 years of experience in preclinical research. As a study director at Oncodesign, Caroline has been working closely with our clients to select the most appropriate study design for the evaluation of the therapies through preclinical studies. Caroline is also involved in InMoD, that is to say Innovative Models Initiative, a French consortium dedicated to the development, characterization and commercialization of new preclinical models in oncology, including pediatrics models and humanized mouse models. She's also the co-author of several publications about PDX. And also to make the webinar a bit more dynamic, so there will be a series of polls. So before Caroline starts, I'm going to ask you a first poll regarding, this webinar. What do you expect by attending this live webinar? So you've got a couple of seconds just to give your answer. So you'll be able to orient it the expertise and the scientific level of the webinar. A few more seconds to go before I close the poll. Off we go. Thank you very much. And now, the floor is to Caroline. So, Caroline, the floor is yours, so you can just carry on with your presentation.
Yeah, thank you, Florence. So good morning and good afternoon to everyone. The topic of this presentation is the use of humanized mice to evaluate immunotherapy in the field of cancer. The main objective is to give you the keys to choose the most appropriate humanized immune system model for the evaluation of your compound. So we'll describe different strategies we adopt here at Oncodesign to humanize the mouse immune system. And the use of these models will be illustrated by various case studies and results. So firstly, to beginning this topic, immunotherapy is one of the most exciting recent advances in the field of cancer treatment. Different approaches are developed, such as cancer vaccines, adoptive cell immunotherapy, or immune checkpoint inhibitors. And a number have been regulatory approved or are currently investigated in clinic as mono or combination immunotherapies. Cancer immunotherapy has given important clinical response, but only in a minority of patients and in defined cancer indications. So effective immunity against cancer involves complex interaction between the tumor, the host, and the environment. So the assessment of cancer immunotherapy requires the use of appropriate preclinical models that reflect the physiological situation in humans. So we have syngenic tumor models that constitute a very useful tool to evaluate new immunotherapies. But one major disadvantage is that they depend on murine immune system, which does not always recapitulate the human immune system and the human immune response. So humanized mouse, referring to a mouse that has a human tumor and a human immune system, constitutes models that are highly relevant to immuno-oncology drug development. So in this first part, I will describe how to generate mice reconstituted with human immune system for immuno-oncology research. Humanized immune system models for research on cancer immunotherapy comprise three elements to define a custom-made preclinical model. So these three elements are the immunodeficient host, the human immune cells, and the human tumor. And like a drawer chest, each element, the host, the human tumor, and the human immune cells, will be selected to define the appropriate humanized immune system model for the evaluation of immuno-oncology therapy as a custom-made study design. So the starting point of the humanized mouse model is the selection of the immunodeficient mouse recipient, which forms the basis of the model, if I can say like that. Here on the left, this is a figure that summarizes briefly the history of the development of immunodeficient mice for humanized mice model. So in retrospect, nude mice and skid mice were the first immunodeficient strains. Other strains were then generated to improve engraftment capacities. And the most significant advance is the creation of NOG, NSG, BRG mice by knocking out the common gamma chain of the IL-2 receptor in not-skid and BALB/c RAG2 null mice. And then progressively, these strains were improved, and most strains that express human cytokine have been developed to support immune cell differentiation and maturation. The latest generation of mice are NOG-XL, NSG-SGM3, and BRG-SF mice. They were developed by CIA, the Jackson Laboratory, and the Pasteur Institute. And then they are now marketed by Taconic, Charles River, and Genoa for this different strain of mice. Today, the mice most commonly used for immuno-oncology research are not-skid, NSG, NOG, NSG-SGM3, NOG-XL, and BRG-SF mice. The second element is the human immune cells. two major sources of human immune cells are currently used: PBMC and hematopoietic stem cells. And we will see later that for the PBMC source, is it possible also to select and use specific immune cells, such as myeloid immune subtypes. And for hematopoietic stem cells, we commonly use here at Oncodesign fresh umbilical cord blood. And the last element is the human tumor. So both human cells and PDX represent relevant preclinical tools for immunotherapy assessment. And various criteria, such as related to tumor molecular characteristics and tumor growth parameters, should be taken into account to select one tumor. So the first approach to humanize the mouse immune system is the use of PBMC samples. So immunodeficient mice, such as Notsky, Danergy, Nog mice, are injected with mature lymphoid population. Human PBMC humanized models develop rapidly from easily available PBMC source, and so provide a perfect model type for short-term studies. T cells are the main immune subpopulation that is present and remains functional in the murine host. This model is mainly used to assess the activity of agent targeting T cells. One limitation of this model is the poor B cell reconstitution. The main disadvantage is the development of graft versus host disease, resulting in a limited treatment window. And additionally, immune system reconstitution and tumor implantation need to be synchronized to ensure that the immune system reconstitution occurs when the tumor reaches the target size, and to ensure to finish the study before GVHD manifestation. Another limitation is donor-to-donor variability. Efficacy can vary in mice from different donors. That's why it is important to use several donors in one study. The other approach is the injection of human hematopoietic stem cells to immunodeficient mice, such as NOG energy, BRGS. For this model, purified human CD34 positive cells from umbilical cord blood are injected to newborn or young adult mice. In comparison with the PBMC, the reconstitution is slower. A successful engraftment is observed around 10-12 weeks of post-engraftment. Engraftment in the hematopoietic stem cell model leads to stable reconstitution of B, T, and K cells, as well as myeloid lineage in latest generation and NSGHM3 and NOGOXL and BRGSF mice. This model can be used for long-term studies. It is the ideal model to assess agents which require multiple lineage immunity and represent and to investigate long-term response. The main limitation is that all human hematopoietic lineage are represented, but not all are completely functional. In particular, B cell maturation is incomplete. T and NK cells display some functional impairments. And hematopoietic stem cell models are more expensive to implement, and studies are longer in comparison with PBMC mouse models. And as observed in PBMC models, donor variability is also described within studies with hematopoietic stem cells. Oncodesign has experience with various tumors in mice reconstituted with human immune cells, as various cell lines as solid and hematological tumors, as well as PDX that are mainly developed in the frameworks of the French consortium named IMODI. And we have here a list of tumors we have already used in humanized mice. Florence, maybe it is time for a second poll?
Yes, this is
Yes.
poll times. So the second one is just to evaluate if you have any forthcoming project related to our current webinars. So we've got a few seconds to go. I mean, to complete the webinar, the poll. Sorry. Okay, a few more seconds to go. Okay, here we go. I'm closing the poll. And this back to Caroline's presentation.
Yeah, okay, thank you. So several examples of the preclinical evaluation of immune-based therapies performed in humanized immune system mice will be presented here. So the first class of immuno-oncology biologics illustrated here is engineered T cells. So that is the simplest humanized models to implement. The humanized immune system models is comprised of NOG or NG mice implanted with a human tumor and injected a few days later with a cellular-based therapy that constitutes the human immune cells. So depending on the origin of the human tumor and the site of implantation, so subcute or IV, the tumor evolution over time in control and treated animals will be assessed by standard method such as calipers for solid tumor. So here on the right, the example on the right, or survival time for disseminated tumor. So the example on the left with the hematotumor. And alternately, a non-invasive method such as imaging can be used to evaluate tumor burden, are illustrated here in the left. A second class of compound illustrated here is T-cell recruiting agents, such as a bispecific antibody. So here, the most appropriate mouse model is that is comprised of NOG or NG mice, humanized with PBMC, and implanted with a tumor expressing the target against which the antibody is directed. In this first case, the tumor model is the simplest, meaning hematological tumor injected by IV route in the animal. So here that is the Ramos tumor, nemato tumor. So immune cells are injected few days later, and they are easily in contact with the tumor cells. And this example, tumor burden, is evaluated by flow cytometry. The proportion of tumor cells is decreased here, is decreased in the bone marrow from treated mice in comparison with control mice. And consequently resulted in an increased survival time. And at the same time, T-cell recruitment is evidenced by in bone marrow from treated mice. Here, it is evaluated by flow cytometry analysis. So here we evaluate the same class of compound using the same humanized mouse models, so meaning PBMC, but with an ovarian tumor injected by IP routes and characterized by acidic and solid masses in the peripheral cavity. So a little bit more complex than hematotumor. Human C125 is a standard biomarker used in the clinic to monitor ovarian cancer in women during and after cancer treatment. So human CA125 was then used as an efficacy parameter to follow ovarian tumor progression in the study. So CA125 levels increase over time in tumor-bearing mice, humanized and non-humanized mice treated with vehicle, as that reflects the tumor development in these mice. The treatment with the bispecific, here in yellow, engager, caused a large reduction of CA125 over time, indicative of a strong anti-tumor effect. And the absence of ascites and macroscopic peritoneal tumor nodules at the necropsy confirms the efficacy of the compound. To make the humanized model more complex, the human tumor is subcutaneously implanted, and the human cells are injected by IV route when the tumor is established, meaning the tumor volume is about 100-200 cubic millimeter. The human cells have to infiltrate the tumors. In both examples here, hematological and solid tumors, the treatment started a few days after PBMC injection, when the tumor reached about 100-200 cubic meter, what I say, and the treatment induced a tumor regression, a large tumor regression. Early approaches in development of new immuno-oncology therapy have primarily targeted T-cell activity. An alternative approach is to target antigen-presenting cells, such as dendritic cells. This approach requires the development of an appropriate human immune system mouse models to evaluate this class of compound. The idea here is to isolate T cells and monocytes from whole blood of the same donor. Monocytes are seeded to obtain dendritic cells. And the first approach is to firstly inject the tumor, then the T cells when the target, the tumor volume is reached, and we inject the dendritic cells where the T cell population is reconstituted. So in this case, circulating T cells increased over time and are present in the tumor. We will see here in the graph. Dendritic cells are observed in the tumor. They are rare events 19 days after injection. Injection of T cells and dendritic cells did not significantly impact the tumor growth. The main limitation of this model is a short window due to body weight loss and graft versus host disease. So a second approach was consequently defined. Dendritic cells are prepared from monocyte and co-ejected with T cells and the tumor cells at the same time. This model is still in progress and would be useful to evaluate compound targeting dendritic cells as CD40 agonist. Another model is currently used to demonstrate anti-tumor effect of molecules targeting antigen-presenting cells. This model comprised BRGSF mice engrafted with hematopoietic stem cells. The results are not available today, but because it is still in evaluation, but we will share the result when this data will be available. So the last class of immuno-oncology therapy described here is immune checkpoint inhibitor. So literature describes that antibody directed against PD-1, CTL4, CD137, for example, have been evaluated is a humanized mouse model using either PBMC or hematopoietic stem cells. Based on these results described in literature, we investigate the evaluation of anti-tumor efficacy of pembrolizumab in NSG-AGM3 mice, humanized with hematopoietic stem cells and implanted with MDA-MB231 tumor. Tumor growth is reduced in pembrolizumab-treated mice with a maximum tumor growth inhibition of 30%. And moreover, flow cytometry analysis done at the end of the experiment on tumor samples evidence the presence of T cells inside the tumor, and proportion of T cells was enhanced in pembro group. The main issue in this mouse strain is that shortly after hematopoietic stem cell engraftment, these mice develop a severe macrophage activation syndrome characterized by a progressive decrease in red blood cell number, reducing the survival time of animals. So in this case here described, we use the PBMC humanized models to evaluate the efficacy of avelumab against the same tumor, MDA-MB231, implanted in nog mice. A moderate tumor inhibition was observed in avelumab-treated mice with a maximum on 22 tumor growth inhibition. And flow cytometry demonstrates the presence of T cells in the tumor, but without any difference between vehicle and avelumab-treated groups. And finally, a PDX was used to investigate the anti-tumor activity of anti-PD-1 agent pembrolizumab in humanized models. So to give us the best chance to being successful and to be close to clinical situation, the idea was to choose a tumor that is highly infiltrate with human cells. This tumor profile is supposed to be more sensitive to therapy. So the first step was to evaluate the tumor infiltrate in PDX implanted in mice, humanized, with either PBMC or hematopoietic stem cells. So here on the left, a list of breast and ovarian PDX for which the human cell infiltration has been evaluated in the originating patient tumor. And based on this analysis, we select one PDX developed from a patient tumor, poorly infiltrate, and one PDX for a patient tumor highly infiltrate. So here in red, one poorly, one highly infiltrate for each cancer indication. These PDX were implanted into nog mice and grafted with PBMC and NogoXL humanized with hematopoietic stem cells. So here on the right. And as done with patient tumors, immune cell infiltration was evaluated by immunohistochemistry in PDX implanted in humanized mice. The results demonstrate that tumor infiltration in mice is correlated with immune cells infiltration in originating patient tumor. The most infiltrate PDX, EMBRE144, was consequently selected for the in vivo drug efficacy study. And we check before starting the evaluation that this tumor highly express PD-L1, as that is the case. So IMBRE 044 was implanted in PBMC NOG mice and NOG EXL mice and treated with vehicle or pembrolizumab. And unfortunately, as you can see here, pembrolizumab therapy did not induce any tumor reduction under this experimental condition. So why immune checkpoint inhibitors such as pembrolizumab do not demonstrate any significant effect in these humanized mouse models as described in literature? Is it attributed to donor-to-donor variability in immune cells used for these reconstituted humanized mouse models? Interaction between tumor cells and immunomodulators inside the tumor microenvironment play a key role in success of the immunotherapy, but are not sufficient. Does it reflect the difference of response between individuals and between cancer indication, as reported in clinic, since not all patients do not respond to immune checkpoint inhibitors? So the development of more appropriate humanized mouse models is important to evaluate immune checkpoint blockade. So maybe we can improve to select the donor according their responsiveness to stimulation inhibition of the target of interest before in vivo injection, and to also maybe to select and to inject PD-1 expressing T cells. Another alternative approach to the use of humanized mouse model for the study of immune checkpoint inhibitor has been the development of humanized target knock-in mice in immunocompetent C57 black cyst mice or BALB/c mice. The major advantage of these mice is that a clinical candidate can be evaluated in these models, and a murine surrogate is not needed. Humanized mouse models have allowed the evaluation of immuno-oncology therapies, including cellular and antibody-based immunotherapies. They can be used as highly relevant models for development of new immuno-oncology therapies in complement of our syngeneic mouse model platform. They constitute useful preclinical tools when surrogate mouse analog antibodies are not available, or when human origin agents are tested. A large panel of PDX models are available for implantation in humanized mice and used in immuno-oncology drug development. One remaining question is why there is difference of response between patient and between cancer indication. There is no unique humanized mouse models that mirrors the cancer patient and the complexity of the cancer microenvironment. Then we have to select the best model to answer a specific question. And some parameters have to be considered for the humanized mouse model selection, such as the mechanism of action of the molecule we test. The mouse host is important, the tumor molecular and the growth characteristics, the type of human cells engrafted and supported, and the advantage and limitation of each humanized mouse model here summarized on the left. And some points that are important to keep in mind for study design is the impact of the humanized immune system on tumor growth and the use of multiple donors due to donor-to-donor variation. So maybe now, Florence, it is time for the last poll.
Yes, Caroline, this is
Yes.
poll times. So the third and the last poll is about the criteria to select a CRO. So you've got a multiple choice. So please go ahead and let us know what will be the criteria that you will choose. Okay, so thank you very much for your participation. I'm closing the poll. So Caroline, I mean, you can carry on with your talk.
Yeah, it's the last slide. So one question is still pending: how to improve the human cell reconstitution that is essential for the assessment of immunotherapies in humanized mouse models. Today we use PDX in our humanized mouse model, and we implant them in mice reconstituted and humanized with either PBMC or hematopoietic stem cells. The heterologous origin of the tumor and immune cells may cause artifacts due to alloreactivity. So in this context, development of PDX models harboring both a human immune system and a tumor fragment from the same patient may lead us towards personalized medicine and may be useful for immuno-oncology drug development. This model development is still in progress in EMODI program. Human-induced pluripotent stem cells technology is used as a source of hematopoietic stem cells. And in literature, more and more scientific publications demonstrate relation between microbiota and cancer, suggesting that modulation of microbiota could improve response to immunotherapy. So the question is, are humanized microbiota immune system models the future for preclinical immunotherapy research? And finally, preclinical models and study design need to reflect the variability of response in patients to be predictive. A variety of PDX models combined with a variety of human donors provide a single mouse trial or clinical mouse trial to assess immunotherapy. Maybe it could be the next step for this type of model to be more predictive. So thank you for your attention.
Okay, thank you very much, Caroline, for this very interesting talk. So now it is the Q&A session. So please go ahead and write your question on the chat box on the right-hand side of your window. So I've got already a first question coming up. So I have. So let me read it out. I mean, to you, Caroline. I've got one T-cell engager molecule. How long will it take to assess my drug efficacy?
Mm-hmm. So it will be depend of the different parameters. The first one, if we choose a tumor, firstly, if it's a PBMC PBMC model or hematopoietic stem cell model, because as we saw, the time to have a humanized model is different. PBMC is shorter. And the second parameter is the tumor models. So it depends if we have already experience in humanized condition, if we have data of the tumor growth in humanized condition, and if it's a rapid tumor growth or a slower tumor growth. So if it's a tumor model we know, we can start directly with the anti-tumor efficacy, and if it's a rapid growth, within 2-3 months we can have for all technical work we can have the data, and we can have one more 1 month and a half before the start of the experiment to define the study design and for administrative part to have the quote to start the experiment. So 3-4 months with a quick PBMC models and rapid tumor growth for the tumor model. And if we choose a tumor model that is slower in terms of tumor growth, so it will take longer to obtain. And I thought in to PDX models we can have some 4 or 5 months to have data because it's a longer tumor growth. But it's important to define the appropriate model depending of these different parameters, and we are available, of course, to design as a customer preclinical model your study design depending of your compound, depending of your question, and to define the most appropriate model.
Okay, so thanks a lot for this very detailed and answer. So I hope the person will get all the information that he or she wants. So I've got another questions coming up. It is regarding the location. So will you plan to collaborate with a US-based service provider?
So we have a team in Montreal in Canada. And they have capabilities to perform such studies using humanized mice. So we are able to perform studies with our team in Montreal.
Okay, so now everybody knows that we've got a subsidiary in North America, so that's very nice to see. Yeah. And they're really ready, just to do all the proper job as if they were in our premises in Dijon or in Paris. So I've got another question. Can you make CD34 plus mice with neonates? Sorry.
Yes, we can perform. We have experience with both neonates and young mice for the humanization with hematopoietic stem cells. So both are possible.
Okay. So thanks for your answer. So please don't hesitate to type your question on the chat box. If you want to. I'm just taking advantage of this couple of minutes just to tell you that by tomorrow you will be receiving an email with a link to be able to see the replay of this session. So don't hesitate, just to have a look and to share with your colleagues. And also, if you have any further questions you don't want to ask here, please feel free to contact us or directly Caroline or myself, or just go to the website and have a live chat with the guys there. They'll be happy to give you all the possible answers. So I've got no further questions. So do you have any more further information, Caroline, you would like to add before we close the webinar session?
No, but don't hesitate to share your project or your and we will be able to advise and to support to define a custom-made preclinical model for your compound, for evaluate your compound.
Okay, so I think that we can close the webinar on all the strategy, just to predict the effect of immunotherapy. So thank you very much for your participation to all the people. Thank you very much, Caroline, for your brilliant talk, and I hope we can see all of you very soon. Thank you very much, and have a nice day or a nice evening. Take care to all of you. Stay home and stay healthy. Bye-bye.
Bye.